Good morning everyone. So today topic is nitrogen cycle and nitrogen fixation. In that nitrogen fixation. So why nitrogen cycle means nitrogen is a macro molecule, and this nitrogen is most abundant most abundant essential mineral elements need for plant for its growth and development. And uses of nitrogen to the plant. Uses of nitrogen to the plant. Usually nitrogen is used for synthesis of all kind of proteins in plant. Also nitrogen uses to the synthesis of 20 types of amino acids to in plant. And nitrogen is also used for the synthesis of nucleic acid like DNA and RNA in plant. And also nitrogen is useful for synthesis of hormones like phytohormones in plant, and also various enzymes and also useful for synthesis of very important chlorophyll pigments. So for synthesis of all these essential organic molecules because of nitrogen. So forms of nitrogen availability to the plant. Forms of nitrogen availability to the plant. Generally nitrogen available in three forms to the plant. There NO3 minus nitrate form. Plant mostly uptake nitrate form, and one more form is the NO2 minus nitrite form. So plant use or uptake nitrate form in very least, and it is the most uptake form of nitrogen is nitrate. Least uptake of nitrogen is nitrite. And even though in ammonium ion form also nitrogen is available, it is moderate. So these are the different forms of nitrogens. Now coming to most important thing is the process of nitrogen cycle or the mechanism of nitrogen cycle. So generally nitrogen cycle which occur in three process. That the first one, nitrogen fixation process. Second one is nitrogen assimilation process. And the third one is most important is nitrogen metabolism. So with these three process, the nitrogen cycle will be occur. In nitrogen fixation, atmospheric nitrogen is converted into ammonia in presence of nitrogenase enzyme. In nitrogen assimilation process, the form ammonium utilized by the plant actually this ammonia converted into ammonium ion, then this ammonium ion later used by the plant for synthesis of amino acid by two process synthesis from ammonium ion. They are reductive amination process. Amination process. Another process is there, that is transamination process. So with these two process, the ammonium ion utilize to synthesis of two types of amino acid by these two processes. So all these processes include nitrogen assimilation. Now coming to nitrogen metabolism, this process includes three major steps. They are ammonification, then one more process, nitrification, and last one is denitrification. So these three major steps are included in nitrogen metabolism. So in this video lecture, we discuss only nitrogen fixation process. Now upcoming videos we will discuss separately as nitrogen assimilation and nitrogen metabolism. Okay, so how nitrogen fixation occur, we'll discuss. So fixation of atmospheric nitrogen. Fixation of atmospheric nitrogen. In atmospheric nitrogen, availability is 78%. Now this fixation of nitrogen, atmospheric nitrogen, occur in two major process. One is without microorganism, the atmospheric nitrogen fixation occur, and with microorganism, atmospheric nitrogen is converted into ammonia. So without microorganism, this process also called biological or physical nitrogen fixation, and with microorganism process called biological nitrogen fixation, or also we can use the term physiological nitrogen fixation. So here this is a biological or physical nitrogen fixation in which without microorganism only atmospheric nitrogen is converted into ammonia. So again this biological or physical nitrogen fixation occur in two processes. One is naturally nitrogen fixation occur. So natural nitrogen fixation, or one more, synthetic nitrogen fixation. Here natural nitrogen fixation occur by electrical nitrogen fixation process, And here synthetic nitrogen fixation occur by industrial nitrogen fixation. Here in biological nitrogen fixation and physiological nitrogen fixation, so here microbes are used. Here microbes only use to fix atmospheric nitrogen, then convert into ammonium ion. The microbes which used in biological nitrogen fixation, such microbes or microorganisms are called diazotropes. Diazotropes microbes. Now first we discuss how electric nitrogen fixation occur. So here, in which electrical nitrogen fixation process, here atmospheric nitrogen combined with atmospheric oxygen in presence of ultraviolet UV rays and during thunderstorming, then it become formed into 2 molecule of nitrogen oxide, and later this 2 molecule of nitrogen oxide combined with water molecule, then it form 2 molecule of nitrogen dioxide and 2 molecule of nitrogen trioxide, and then this 2 molecule of nitrogen dioxide again combined with water molecule. When it combined with water molecule, then it form HNO2, here nitrous acid, and 2 molecule of NO3. When it combined with water molecule, then it form HNO3, nitric acids. And this nitrous oxide and nitric oxide both are formed in rain in the form of acid. So acid rain. So this acid rain, when reached to the soil, in soil the acid rain form of water in the form of HNO2 and HNO3 dissociation into NO2 minus nitrite and nitrate form. So in this form the plant utilize the nitrogen. So this is all about the natural nitrogen fixation in the electrical nitrogen fixation. Now coming to synthetic nitrogen fixation by industrial nitrogen. So here what happened, in which nitrogen combined with 3 molecule of hydrogen at 450 degrees Celsius, then it to become form 2 molecule of ammonia, and now this two formed 2 molecule of ammonia combined with carbon monoxide, then it become form into urea, NH2CO NH. So this is what urea. So this formation of ammonia in industrial level is called by a process called Haber-Bosch process. So Haber-Bosch process, there is a formation of ammonia by nitrogen and hydrogen in industrial level, and this ammonia later combined with carbon monoxide to get formed into urea. So when we use urea in agriculture in field, then this urea again come back and it is available to the plant in the form of ammonium ion. So this is how plant utilize atmospheric nitrogen in different forms. Here use atmospheric nitrogen in nitrate form. Here plant utilize atmospheric nitrogen in the form of ammonium ion. Now we will discuss how microorganisms involved in atmospheric nitrogen fixation process. The microbes which involved in atmospheric nitrogen fixation process, they are called diazotropic microbes. Why this name means, because of this only these microbes have special enzymes. These microbes only contain special enzymes. So these enzymes are called nitrogenase enzyme. Nitrogenase enzyme is the most important enzyme. This enzyme play crucial role in atmospheric nitrogen fixation process. And this nitrogenase enzyme, we will discuss some important things about nitrogenase enzyme. So generally these nitrogenase enzymes are made up of with molybdenum iron protein compound. Molybdenum and iron protein compound structure it is. And next one, it helps in, this nitrogenase enzyme helps in conversion of atmospheric nitrogen into ammonium ion. And one more important thing is, there is a gene which is essential for synthesis of nitrogenase enzyme, that is NIF gene, NIF. So NIF gene is essential for the synthesis of nitrogenase enzyme. And the most important thing is, this enzyme is extremely sensitive. This enzyme is extremely sensitive to oxygen. Hence there is a oxygen scavenger. To protect nitrogenase enzyme from oxygen, there was a protein which is formed and it acts as an oxygen scavenger, that is leg hemoglobin. So this leg hemoglobin is act as an oxygen scavenger. So protect the nitrogenase enzyme from the oxygen. And this leg hemoglobin not only act as an oxygen scavenger, but also it gives pink color to the nodules. And here leg hemoglobin is a protein which is formed by two organisms. It means that it is, here I am writing leg hemoglobin formed by plant pot and microbes. Here microbe is bacteria. Here most important bacteria is rhizobium. Here most important plant is Fabaceae plant or leguminous plant. Here in this leguminous plant form protein part, that is globin and rhizobium bacteria form heme part. It is a iron compound part. So like that, globin and heme both together form leg hemoglobin. So it means that leg hemoglobin is not only product of any single organism, it is a product of both plant and bacteria. So this is all about the leg hemoglobin. Now coming to one more important things about nitrogenase enzyme, how this nitrogenase enzyme consumption of energy, how energy will be consumption by the nitrogenase enzyme. So we will discuss here. Consumption of energy by nitrogenase enzyme. Here, to formation of two molecule of ammonium, 16 ATPs get required. It means that the here formula is atmospheric nitrogen combined with 8 proton plus 8 electron plus 16 ATPs in presence of this nitrogenase enzyme, then it make into 2 molecule of ammonia, and here hydrogen plus 16 ADP and 16 inorganic phosphate. So remember, to formation of 2 molecule of ammonia, there is a need of 16 ATP. And one more important points you need to remember, for fixation of for formation of 1 molecule of ammonia, then it required only 8 ATP. So here, first one, if 2 molecule of ammonia will get formed, 16 ATPs are required. For formation of 1 molecule of ammonia, 8 ATPs are required. So, but in exam, they will trap regarding to this ATP. So, you need to remember, if in exam, if they ask for one molecule of ammonia formation, the required energy is 8 ATP. If in exam they will ask for fixation of nitrogen, how many ATPs are required, means 16 ATPs. So here you will get some confusion, but thing is, the for fixation of nitrogen means here only nitrogen, one nitrogen get fixes means how many amino ammonium ion form, means 2 molecule. So if 2 molecule of ammonium get formed, means how many energy we required, 16. For here asking for 1 molecule of formation of ammonia, we need only 8 ATP. But in this here, for fixation of 1 molecule of nitrogen, we required 16 ATP. It means that we have to only understand that for fixation of 1 nitrogen, how many ammonium will be formed. So that we won't get any confusion by these two questions. So this is all about the consumption of energy by nitrogenase enzymes. Now we will discuss how biological nitrogen fixation and what are the microbes they use in biological nitrogen fixation. We will discuss in detail. Here, so biological nitrogen fixers microbes. Biological nitrogen fixer microbes are also called diazotropes microbes. So here you need to remember, so what is the main function of these microbes? This microbes helps in conversion of atmospheric nitrogen into 2 molecule of ammonia. Right. So we classify these microbes into 2 categories because of there are some microbes that are freely living and some microbes are mutually or symbiotically living. So here first we discuss free living microbes. Free living microbes. And here symbiotical microbes. Here free living microbes, again we can classify them into 2 categories because of there are some microbes they need oxygen for their survival, and there are some free living microbes in absence of oxygen they can survive. So based on that, we again subcategorized into 2 groups. They are aerobic free living nitrogen fixer and anaerobic free living nitrogen fixer. So here aerobic means in presence of oxygen, anaerobic means in absence of oxygen. So coming to example for aerobic free living nitrogen fixers are Azotobacter, and one more important is Bizirinke. Here. So these two microbes comes under aerobic free living nitrogen fixers. Here Azotobacter, it is a PPLO bacteria. PPLO bacteria means pleomorphic in nature. So here PPLO means pleuropneumonia-like organism. It means that the bacteria, it can change its shapes and structure depend on type of environment and type of mode of nutrition. Such bacteria are called pleomorphic bacteria. The best example is here Azotobacter, and already we discussed, already we made one video in mycoplasma. Even mycoplasma is also one kind of PPLO bacteria. Now coming to anaerobic free living nitrogen fixers. Best examples are Bacillus, and one more important, Rhodospirillum. So these two microbes comes under anaerobic nitrogen fixers. Now coming to symbiotic microbes. So these microbes mutually beneficial to each other, and here we can classify them two category. Because of these microbes can form nodules, and some microbes they won't form any nodules, still they symbiotically associate with the plant. So based on that we classify them into two categories. So with nodule formation, and here without nodule formation. Here again the place where the nodule will be formed. Based on that we again categorize into subgroup. Because of nodules mostly usually form in root, but in some microbes they form nodules in stem also. So based on that we classify them into two categories. So like root nodule and stem nodule. In root nodule again we classified into two groups because of there are the microbes which form nodules in usually in leguminous plant. So other than leguminous plant, the root nodules also form in root like non-leguminous plant. It means that so not only leguminous plant the nodules are formed in root, but there are some plants like example Frankia. So in that nodules are formed in root, but it is a non-leguminous plant. So based on that we again divide into two. So here in root nodules in leguminous plant. Here leguminous plant means Fabaceae plants, and here non-leguminous plant. So leguminous plant nothing but they are Fabaceae plant like pea plant, bean plant, and alpha, etc. So in this plant the microbe is rhizobium, very important. So rhizobium is a microbe which form nodules in root in this leguminous plant. Now coming to non-leguminous plants. So example plants are ulnus plant and casuarina, very important plant, and myrica plant. So in this plant and this plant are generally non-leguminous plant. So in this plant there is a one microbes which form nodules in root. The microbe is Frankia. So Frankia is a microorganism, belongs to actinomycetes group. Actinomycetes group. Actinomycetes group means it is a filamentous bacteria. So this actinomycetes are filamentous bacteria which form uh, root nodules in Alnus, Casuarina america, like non-leguminous. Now coming to stem nodules. So there are one plants in which the nodules form in stem. The plant is Sesbonia plant. So here the microbes which form nodules in Sesbonia form is Azorhizobium. Azorhizobium. So here don't confuse. Here we have Azotobacter is different. Azorhizobium is different. Azorhizobium form stem nodule in Sesbonia plant. Now coming to without nodule formation, and means so there is no forms of any kind of nodules. Still the microbes can symbiotically associate with the microbes. For example, here there is a one bryophyte plants called Anthocyrus. Anthocyrus is a bryophyte plants. So in which there is a one blue-green algae or cyanobacteria can symbiotically associated. The blue-green algae name is nostoc. So nostoc is a microbe is symbiotically associated with the bryophyte plant, that is Anthocyrus, without nodule formation. And there is a one more plant is there, but this plant is belongs to pteridophyte, that is Azolla plant. Azolla, it is a water fern plant. It is an aquatic water fern plant. Azolla belongs to pteridophyte plant. And with this plant, there is a one more cyanobacteria or blue-green algae form symbiotical association with Azolla. So that is Anabina. So Anabina is a blue-green algae. And very important in one more important plant group, that is gymnosperm. So in gymnosperm one plant is Cycas. In Cycas we have corolloid root. So in corolloid root only, corolloid root of Cycas, and there are two blue-green algae, they form symbiotical association. They are nostoc and Anabina. So both blue-green algae are also called cyanobacteria. They form symbiotical association with the corolloid root of Cycas. So this is all about the biological nitrogen fixer microbes. How biological nitrogen fixers microbes are used, in which convert atmospheric nitrogen into ammonia. So with this we have covered the entire concepts so in this video. So now it's time for you here. So here I have given 3 highly important NCERT based MCQs on this screen. Here I will show you. So this is the first 2 questions. See, consider the following statement regarding the free living nitrogen fixing microbes. So here there are 3 statements are there, 1, 2, 3, and which of the statement given above are correct. So here these are the before attempting these questions, and so once you go through this video and drop your answer in comment section. And one more important MCQ is second one here. Here we have second question. Consider the following statement regarding the enzyme nitrosinase. Here also we have 3 statements. So select here which of the statement given above is correct. And one more highly important NCERT based question here. Third one here. Here third one. During the symbiotic nitrogen fixation in legume nodules. So what is the primary function of the pigment called leg hemoglobin? So just now we have discussed about leg hemoglobin. So read it, four options, and click it and do the right answer in the comment box. So this is all about the nitrogen fixation, in which nitrogen fixation we just cover only one process, that is nitrogen fixation. For upcoming videos we'll make another videos on nitrogen assimilation, and another video we can make